Okay, now we're back, picking up where we left off. Now we want to talk about cancer and the cell cycle, and why is the cell cycle important when we talk about cancer. And it is because, remember those stops that we talked about earlier, those checkpoints when the cell cycle goes unmonitored or uncontrolled. Essentially, when we lose control of the cell cycle, that's when cancer has an ability to develop. So, cancer is essentially uncontrolled mitosis. We're not able to control these events that are taking place in the cell cycle. So, um, we've got to understand, mitosis in order to be able to make way with this thing that we call cancer. So there are five main categories of cancer. You will not be tested on this. This is just useful information. The five main categories. Carcinomas, these guys begin in the skin or tissues that line organs. So usually you're talking about your epithelial tissues, okay? The skin is made up of what we call squamous epithelial tissues. These cells are flat, okay? And then the same tissues that line our organs, like blood vessels, those are also called epithelial type tissues. And then we have sarcomas. Those are ones that develop in things like our connective tissues or muscles. So bone, cartilage, fat, and then muscle, again, or other connective tissues. And then we have things like leukemia, which develop in the blood and bone marrow. And you know that with a leukemia, you'd never have a mass. So this is one that does not form tumors. And then the lymphomas. These start in the immune system. If you remember, we have what's called a lymphatic system, which is part of our immune system. This is the cleanup portion of the body that rids the body of all sorts of things that could do it harm. And then the central nervous system, where you can have cancers that develop in the brain and the spinal cord. So the word carcinogenesis means the development of cancer. Carcinogenesis means the development of cancers. Now, cells have been known to have a programmed number of times that they actually divide, which means that, what we're talking is they divide, they make copies of the cells, then they divide again and make copies. Well, cells have a set number or program number of times that they divide, and then they are programmed for cell death. So we can say some of the cells, and we're not talking about all of them, but just as an example, repeat the cell cycle 50 times, and then they are programmed for programmed cell death, and which is also apoptosis. But we know that cancer cells are immortal. They live on and on and on and on. So one of the papers that I'm going to have you write has something to do with some cells that are still alive that were harvested back in the 50s. Amazing cells they were. But anyway, cancer cells, again, we said they're immortal, and they are able to, and we're going to talk about this later, how they are able to disrupt, how they're able to displace and destroy healthy cells. So they're able to disrupt, displace, as well as destroy healthy cells. Now, to understand cancer, I want to bring to your attention there are four major classes of genes that are often implicated in the origin of cancer, and that's because these are the most commonly mutated genes. And those genes that I want to bring to your attention are proto-oncogenes, tumor suppressor genes, genes that control apoptosis, and genes that are used to detect as well as repair DNA. Genes that are used to detect and repair DNA. So let's start with the first gene, which is going to be our proto-oncogene. Now, proto-oncogenes, these genes make proteins, because if you remember when we talked about a gene back in chapter 4, we said when genes are expressed, there's some type of protein. That protein might be a structural molecule, it might be a signal, it might be a receptor, it might be many different things. But now we're talking about a signal, okay? So this proto-oncogene creates a protein, and that protein signals normal growth, cell growth, as well as division. But if that gene, which we know are made up of DNA molecules, have been mutated, so in other words, we don't have the same coding in the base pairs that we had before, then that's going to tell us that something is wrong with our DNA. Those changes in the DNA are called mutations. And when those mutations occur, they may change the product that's actually being produced. So the signal that once told us to proceed with normal cell growth may be turned on uncontrollably. When these cells are turned on at a high express rate, then generally we call them oncogenes, okay? So when they're expressed at abnormally high levels or rates, we call them oncogenes. And you know, once we have an oncogene, then you're going to need a who? An oncologist, okay? But anyway, normally these genes start, again, we said normal cell growth, but when they're mutated or changed, then they can be turned on at excessive rates. All right. Then the second set of genes, we said these are the genes that work basically hand in hand with the proto-oncogene. So once enough cell growth is taking place, this is the gene that will be expressing its product to say, okay, it's time to stop growing. But again, these genes can be mutated. Mutated from what? Just because of your gene pool itself, or mutated because of whatever you were exposed to environmentally, or the x-rays that the doctors ordered for you, or the things that we eat, the things that we smoke, the things that are just in the air that we breathe, or the, you know, so the list goes on and on and on of those things that we're exposed to. So it says when they are mutated, these prevent the tumor suppressor genes from functioning. So then what can happen? If the tumor suppressor genes are not producing the product that says to stop growing, then these cells can still continue to make copies and make copies and make copies. Okay? So again, we want to be reminded that these genes normally inhibit cell division, which help to maintain that balance with the proto-oncogene. Our next gene then is the genes that control apoptosis. All right, so again, the product says that's going to be produced is going to say, hey, this cell has divided ever how many times, and now it's time for it to commit suicide, basically. Well, if there is a mutation in this gene, again, we're going to lose control of the cell cycle, and these cells are going to continually be made, and because we have now developed, because of the mutation, an abnormal resistance to programmed cell death. Again, this can also contribute to the development of cancer. Now, our final class of genes, again, are the genes that detect and repair DNA. Remember when we were talking about those checkpoints, and we said it was very important that we stop, we take a look at the DNA, because we didn't want to keep making bad copies of DNA, because bad copies of DNA can lead to the development of cancer. So it says we want to be able to detect those problems, and then but we also want to be able to do what? Repair. But if there's damage or mutation to the gene that does this, or the several genes that are responsible for controlling this function, then we've lost that control. So we end up with cells that have DNA that have a higher rate of mutations because we were not able to fix the mutations that had already occurred. And we know that this can also contribute to cancer. One of the breast cancer genes has this issue, not being able to detect and repair the DNA. So the BRCA1 gene is in this class of genes that are responsible for detecting and repairing DNA. Now, very important tumor suppressor gene. So moving on. So, and by the way, that sounds like a great essay questions. The 4 genes that are implicated in the origin of cancer. You should be able to discuss what they normally do and what happens if they are mutated. Now, this is an example of one of those genes that we discussed. The second type of gene that we discussed were the tumor suppressor genes. Well, this is a specific tumor suppressor gene. This gene is called the P53 gene. Most organisms who are said to not have this gene are said to be genetically unstable because this gene is the gene that normally is responsible for putting on what's called an emergency brake. So this gene is also known as what? The guardian of our genome. What is the genome? That's our genetic information, our DNA, our genes. And so this P53 gene usually functions to properly control the cell cycle. Because remember we said uncontrolled mitosis is what's going to lead to the development of cancers. So again, we said this guy will act as an emergency brake if there's something wrong with the DNA, inducing either arrest, hey, let's stop, let's get these mutations under control, let's try to repair it. But if we cannot repair it, then we want to send the cell into apoptosis. And this will help us from accumulating those excess mutations and passing them along from one cell to the next cell. All right, so now let's talk about the common characteristics of cancer cells. Well, now we want to focus on the common characteristics of cancer cells. Now, again, another possible essay. These are excellent topics for multiple choice, but more so a written exercise in the form of an essay. So the common characteristics of cancer cells. These guys have an abnormal nuclei. We could say they are increased in size, having too many chromosomes. They lack differentiation. They are able to evade your immune system. They have a loss of contact inhibition. We're going to talk about what every last one of these mean. And they are able to stimulate angiogenesis. And then we said they also can round up, break attachments, become metastatic, and move elsewhere and set up a tumor distant from that original tumor that we had. So increased size. What are we saying with increased size? They have an abnormal nuclei. Mayo Clinic has said that these cells that have too many chromosomes may lead to the development of cancer. So they have a large nuclei, often having this abnormal number of chromosomes. If you go back to the first slide, look at the nuclei of those cells in comparison to the some of the nicer cells that are around it. They look as if there's something wrong. All right, so again, we said normally damaged cells undergo apoptosis, but cancer cells do not. The next topic is cancer cells lack differentiation. What do we mean by differentiation? We mean that They don't contribute to the functioning of any body part. They don't become bone. They're not specialized for specific tasks that need to take place in the body. And we'll pick up on this in just a little bit.